Welcome to the summary video for the regulating substances chapter. This video will cover video number 1 to number 8, and the second video will cover number 9 to number 16. So I'm going to start with the first dot point, which was explain why the concentrations of water in cells should be maintained within a narrow range for optimal function. Remember, there was two reasons. The first one was that water was for chemical reactions to occur, so water was really important for chemical reactions to occur. So you can imagine this to be the cell, and the blue thing, the enzyme, the red thing, the substrate. For chemical reactions to occur, these two need to come together. Basically, all chemical reactions in our body need to have an enzyme for it to work properly. And if there's no water, they'll be stationary. They're big objects. They're not going to move much without water being present. So without water, nothing moves, whereas if we have water as a solvent, you can imagine these will now swim around and eventually they will meet. So eventually these come together. When they come together, they have new product forming. So then a chemical reaction has occurred. So you have, these are the products. So it's essential, water is essential for chemical reactions to occur. Without water, there are basically no chemical reactions. Without enzymes, there won't be any chemical reactions. So without water, there won't be any chemical reactions either. Optimal levels of water to maintain shapes. The second point was we need to maintain that isotonic shape. A isotonic solution gives us that perfect shape, that round shape, which is good for our cells because now they can function properly. Whereas if we go too low or too high, in terms of our water concentrations, they will either shrivel up or burst, which obviously both of them are not good for optimum function. So water is important for chemical reactions to occur, and water is important for the cells to maintain their good optimum shape. Also, now this one is explain why the removal of waste is essential for the continued metabolic activity. So that word metabolic activity was just how fast enzymes can work and how fast chemical reactions occur. And remember nitrogenous waste, that was urea, and urea can build up in the body if it doesn't get removed. So just imagine these cells are producing nitrogenous waste because they're just breaking down amino acids and protein. But now you have a huge high solute inside the cell because urea is a solute as well, and low solute outside because we are not removing them, we're storing them. So what happens with water? Water will travel from the outside to the inside because osmosis is from low solute to high solute. So this cell will actually burst because it will accumulate too much water. Right? So is that good for metabolic activity? If it's bursting, probably not. So that's one reason why we need to remove waste to maintain an isotonic environment for the cell to work properly. The other one was to maintain optimum pH. Remember how all enzymes have their optimum pH? For the most of these enzymes, it happens to be about around 7. So if we go too low or too high, so too much urea will mean it's going to go too high, or too much carbon dioxide will mean it's going to go too low. Either of those cases means our enzymes are going to get denatured, and the normal function is impaired, right? So this is denatured, now it's impaired. Normal is what we want to keep, and that's the optimum pH. If it's too low or too high, enzymes become denatured, and chemical reactions don't go as fast as they should. So if chemical reactions don't go as fast as they should, the metabolic activity decreases as well. Now we have the role of the excretory system. Remember mammals, it was two things, both to remove nitrogenous waste, and that was in the form of urea, but also to balance salt and water levels. So those two, and balancing salt and the water levels is what we call osmoregulation. So that was a word for it. Whereas in fish, it is important to balance salt and water levels, but it's not important to remove nitrogenous waste because they can remove nitrogenous waste through their gills. So it's their gills, not their kidneys, that remove nitrogenous waste. So for mammals, it was to remove urea and to balance salt and water levels. For fish, it was only to balance salt and water levels. That was the role of the kidneys. Explain why the process of diffusion and osmosis are inadequate in removing dissolved nitrogenous waste in some organisms. So remember, diffusion was from high concentration to low concentration. So if you imagine this to be the blood, and it's to be the kidney, the Bowman's capsule, the first part is the kidney, then yes, you will have some of these yellow molecules, which are meant to be urea. You will have some of them going over. The problem is this diffusion was too slow, so you don't have all of them going over, and we want to get rid of most of them or all of them. So that was one problem, it was not fast enough. The other problem with diffusion was that it was not selective enough. So that means that because diffusion works on pretty much anything, it's also going to work on glucose and amino acids in this case. So the purple one was amino acids and the blue one was supposed to be glucose. So you can imagine that not just urea goes over, but also your glucose and also your amino acids, and we want to have some way that we can bring them back, and diffusion doesn't allow us to do that. And also, why is osmosis not enough? So osmosis deals with water, so water travels from low solute to high solute. And why is it not enough to remove nitrogenous waste? Well, you can imagine these yellow dots, they won't go over for osmosis because osmosis only deals with water. So osmosis by itself is definitely not enough to remove urea. So these are the reasons why diffusion and osmosis are not enough. And obviously filtration and active transport, we need to have those two mechanisms to actually make both of these things happen. So to remove all the urea and to reabsorb all the good stuff. But I'll go over that in a second, what they are. So they distinguish between active and passive transport and relate these to the process occurring in the mammalian kidneys. So first, distinguish between passive and active transport. Passive transport was something that requires no energy. So it requires no energy. So for example, diffusion, which goes from high concentration to low concentration. Filtration, which is a high pressure movement of substances across a sieve-like layer. So this is where you have that hose just pushing anything that is small enough through that layer. That happens in Bowman's capsule. Got osmosis, which is water traveling from a low solute concentration to a high solute concentration. So all these three are passive transports, so they don't require energy. The active transport is the pumping action, so it's pumping from a low solute to high solute concentration. So that's the reverse of diffusion, but that requires energy, so that was the difference. And don't mix up osmosis and active transport. Remember, osmosis was from a low solute to high solute, water traveling from low solute to high solute. So osmosis only deals with water, and it's from water traveling from low solute to high solute, whereas the active transport that deals with anything, and it's that thing that is being dealt with going from low concentration to high concentration. So active transport is the reverse of diffusion. And where do they occur? Filtration occurs at the Bowman's capsule, so everything is being forced into it. Active transport happens at the proximal and distal tubules, so at the tubules. And active transport was when you have all those good nutrients, the stuff we want to keep, when they get reabsorbed and not lost. That's because we want to keep amino acids and glucose because they're really good for us. We have osmosis. Osmosis basically occurs at all points of the nephron, at the proximal tube, at the loop of Henle, and in the distal tube as well. So osmosis occurs at all parts except for the Bowman's capsule. But apart from that, at all different parts of the nephron. And diffusion occurs with salts, and that is when at the loop of Henle. So when they diffuse back over into the medulla. Salts here. So that was that. We have the kidney dissection. So you might have looked at the kidney in class. And what you've noticed is you would have had these first, I'm going to go over what these two layers are, the cortex and the medulla. And you don't need to know this for the dot point, but it's interesting to know where the nephrons are. So you can imagine this red part here is the same as this red part here. So that's the cortex. And the cortex is where we have our Bowman's capsule and our tubules. And then medulla is this part in here, the blue here, which is the same as that blue here. And that's our medulla. So, and that's where we have the loop of Henle. So the nephron starts in the cortex, goes all the way down to medulla. So you can imagine you have like a whole nephron lined up from here to here, but then you have them, millions of them covering the whole of the kidney. They produce urine, and that urine gets collected in the collecting duct, which is this part here. So you need to identify the regions involved in the excretion of waste products. So these are the regions. You've got the collecting duct, which collects the urine from the nephrons. So when it's produced, it gets collected by the collecting duct. Then the collecting duct dumps it into, so urine is emptied into the renal pelvis from the collecting ducts. So the collecting ducts collected from the nephrons. They send it down to this greenish area here, this part, and there it dumps into. So this is where it's dumped into. So this is like a huge pool of urine, and from there it goes to ureter. Ureter is this part. Imagine a bit like a tube which then connects. So we've got here the kidneys. The kidneys are connected to the bladder via these ureter tubes. So the tube that transports urine from pelvis to bladder, that's our ureter. And when it's at our bladder, eventually it's going to go and be removed. So this is urine and us removing urine. So these are the parts that are part of the excretion of waste areas. Identify the areas on the kidney: collecting duct, renal pelvis, and ureter. And renal artery and renal vein, these are just the veins and arteries that go and bring blood to the kidney and bring blood away from the kidney as well. We've got explain how the process of filtration and reabsorption in mammalian nephrons regulate body fluid composition. So remember, filtration was here. Filtration makes urea go from blood to the Bowman's capsule, so into the kidney. So all those yellow dots are urea, and filtration makes sure that all of them go over. So not just one, but all of them go over. Now it's filtration. Active reabsorption, so what does reabsorption do? So reabsorption does two things. First of all, active reabsorption, so the use of energy, makes sure that nutrients, so I wrote nutrients in two different colors because green can stand for glucose and purple can stand for amino acids. So there might be a low concentration in our actual kidney and a high concentration in the blood vessels around it, but we still want to make sure that we get all of them. So what we can do is we can just make sure we pump all of them over, but we pump them over for active reabsorption, so that costs energy because it goes from low concentration to high concentration. That's one role of reabsorption, make sure that our body fluids are back in high, has all that glucose and all those amino acids back in our blood, because we don't want to lose them in our urine. But also reabsorption helps us to deal with salt and water levels to balance up fluid composition. So if you have too much water, we would get rid of more water in our urine. If you have too little water, we'll conserve water by reabsorbing water. So reabsorption of salts and water deals with making sure that we have that ideal optimum water levels. So if we have too much salt and too much water, we get rid of it. If we have too little, we conserve it by reabsorbing it. Right? So those three things allow us to have that perfect balance of amino acids, glucose, and water and salt in our body fluids, in our blood, because the kidney will make sure anything that's too much, it'll get rid of. And the urea, which is bad in general, it'll get rid of it, most of it it can. Right? So those three processes help us to make sure that our blood is nice and clean and has everything that it needs. Renal dialysis. Renal dialysis was for people who had kidney failure or kidney problems, and they had to go on this machine. Now you have to compare the process of renal dialysis with the function of the kidney. How are they similar and how are they different? So first, similarities. Similarities are that they filter out drugs and poisons. So just like our kidney, that renal dialysis machine also does that. It removes drugs and poisons. It also removes urea, which is good. And it helps us balance our water and salt levels. So the main functions of the kidney, the renal dialysis machine does too, which is good because otherwise people who would have had a kidney failure would die quite quickly, because this needs to happen on a daily basis, or on a regular basis, not on a daily basis. The differences are that diffusion. So with the renal dialysis, we have diffusion only, whereas we have active transport and filtration and diffusion happening and osmosis when it comes to the kidney itself, right? So diffusion only in the renal dialysis machine, whereas we have more mechanisms in the kidney itself. Also, it's much slower. It takes quite some time, 3 hours at a time. You have to be in a hospital hooked up for this to happen, compared to the kidneys, which do that quite fast. And also, it doesn't use hormones, so it doesn't use ADH hormone or aldosterone, whereas obviously the kidney does. So yeah, remember all this, and then we're going to go over the next 8 in the next video in a second as well.